So we can start for the next presentation. So I want to introduce Robert Young. He is an NMR expert at EMSL at PNNL, and now he will talk about NMR-based metabolomics research and how we can do this here for the synthetic biology or in general. Robert, it's your turn.
All right, thank you. All right, so it's an outline of the talk. We'll start out with a brief history, do some basic theory, experiments, get into the workflow, databases, and profiling approaches. I'll show a quick example of how we profile here, typically at EMSL using the A pseudoturris sample that was showcased in the first talk. And then I'll finish with some highlights of select applications from the literature that showcase some strengths of NMR. So nuclear magnetic resonance was first detected in molecular beings by Isabel Rabi in 1938, and he received the Nobel Prize for that in 1924. The first NMR of liquids and solids was performed by the research groups of Edward Purcell and Felix Bloch, and they did so independently in the same year, 1946, and both shared the Nobel Prize in 1952. That was the same year the first commercial NMR spectrometer hit the market. Now, the first application of NMR to study metabolism is credited to Wilson et al., who were studying ethanol metabolism in rats by carbon-13 isotope tracer studies. The field has grown since, and even though for many years, the number of publications kind of hovered at a low level, as you can see, this century metabolomics research involving NMR has really taken off and showing no signs of slowing. That's thanks in part to a lot of advances in techniques and instrumentation that have come out since. So some of the strengths and weaknesses of NMR-based metabolomics, its key strengths include that it's a highly reproducible method. There's no real batch effects associated with it. It's inherently quantitative. It often requires minimal sample prep and no chemical derivatization. It's non-destructive. It's information rich. We use it to do de novo 3D structural elucidation of molecules, and it's amenable to automated workflows. Some of its weaknesses, the biggest one is it's relatively less sensitive than other techniques. It's about 10 to 100 times less sensitive than mass spec, for example. Also, in terms of identifying metabolites, NMR spectral libraries are much less extensive than those in mass spec. Plus, some things that complicate identifications include pH, ionic strength, temperature, and other matrix effects, because they can cause certain peaks to shift. And if your sample contains many metabolites in a 1D spectrum, that can cause a lot of signal overlap and again complicate analysis. So what is NMR? So NMR capitalizes on the properties that certain nuclei possess. That is a magnetic moment that's due to circulating nuclear charge. Not all nuclei are NMR active, but every element on the periodic table except cerium has at least one NMR active isotope. NMR active doesn't necessarily mean NMR convenient or really feasible to study. But fortunately, many of the ones that we like to interrogate in metabolomics are represented by receptive nuclei for hydrogen 1, or the proton, and carbon, nitrogen, fluorine, and phosphorus all have NMR active isotopes that happen to be spin 1/2, which is the easiest case. So what happens in NMR with these magnetic moments is if you put this in a strong external magnetic field, they'll experience a torque and begin to precess at a characteristic frequency. And it depends on this constant here, which is isotope specific. And so it's different for every NMR active nuclei. Additionally, the otherwise degenerate spin states for these nuclei is split when it gets put in a strong magnetic field, and it depends on the orientation of these magnetic moments in the field. Now, this energy difference is really, really small, and so that an ensemble of nuclei in these magnetic fields. The actual population, the number of nuclei with that are in the lower energy state is quite small. And the sensitivity of the NMR experiment is actually proportional to this population difference. And this is why it's one of the least sensitive techniques relative to others. Also, since we can't change fundamental physical constants, as you can see here, we can increase this energy gap by increasing magnetic field strengths. And so there's always a push to keep going up in magnetic field. Now, for our purpose, the proton is the most sensitive stable isotope. It's 100% abundant and it's everywhere. And I've listed some select parameters of spin 1/2 nuclei that are relevant to our research. And also wanted to highlight the fact too that when you hear people refer to their NMR spectrometers, and you hear them call out 500, 600, 800, they're referring to the low frequency of the proton at a particular field, as opposed to just calling it out in terms of Tesla or Gauss. And so in the simplest NMR experiment, the 1D pulse nuclear experiment, we can imagine these nuclear spins, this ensemble of nuclear spins, they're all precessing at the low frequency. And their magnetic moments are randomly oriented, but if you were to add up the component that's in line with the field, what you would get is a net magnetization that's in line with it, corresponding to those in the lower energy state. To probe NMR, what we do is we apply a radio frequency pulse that's resonant with these nuclei. And so we're talking about that low frequency range. We apply this field 90 degrees to the static field, which causes all these spins now to precess about that field. So they absorb the energy, they go through transitions, and now what we have is this net magnetization vector transverse or 90 degrees to the applied field. And these spins are also now all precessing around that field, and so they're coherent. Then that pulse goes off and they begin to precess around the B0 field again. Now we have a detection coil set up, and so these spinning magnets induce a current into these detection coils. And we pick up a signal, oscillating amplitude versus time that corresponds to all these frequencies. This signal decays because relaxation processes cause that phase coherence to be lost over time. And the spins return that energy back to the surroundings and return to the state before we pulse. Now in practice, what we do is we wait until a significant portion of that magnetization has returned. We repeat the experiment, and then we add the results to increase sensitivity. And those are called, each time we do that, it's called transient. Some people still call them scans. We use a Fourier transform to convert this time domain signal into a spectrum at the frequencies of all the constituent nuclei in there. Now,
I'm sorry to interrupt. It seems like your audio seems a little bit quiet. Some people are having a hard time hearing you. By chance, do you have like a headset or something else you can use as a microphone?
I do not. I do not have a headset. I'll try and move this a little closer. I don't know if that helps.
Yeah, it still seemed a little bit quiet. I guess one other option. Do you happen to have a cell phone nearby that you could join for audio?
I do have a cell phone.
Okay, yeah, it'll just take a minute. So if you go to where you can mute and unmute in Zoom and click the little up arrow, there is a switch to phone audio option. And it'll just give you a phone number to call and then prompt you to enter that meeting ID and your participant ID, and then we should be able to hear you a little better.
Okay. Yeah, I'm really sorry about this. I thought in the testing the volume was coming through.
Yep, no problem.
Okay, how am I coming through now?
Okay, I can hear you good, and it sounds like there might have been a little feedback earlier, and I think that might be solved now. So I think you're good to go.
Okay, well, let me know if I get quiet again, and we'll go to plan B. All right, so I'm not exactly sure where I left off. I think what I wanted to say was if all these spins only precessed at the Larmor frequency, NMR wouldn't be very interesting. But the fact is they actually experience slightly different fields based on their location in molecules due to the electron density, because these magnetic fields induce, they get the electrons circulating, and moving charges make magnetic fields. And these fields, they can either reinforce the static field, and that's called deshielding, or they can counter it, and we call that shielding. And these cause the frequencies to change based on its location in a molecule. Now, the transformed NMR spectrum will contain all these different frequencies, and because nobody has a magnet with the exact same frequency to the level of precision that would be needed for direct comparison, we normalize these frequencies to the chemical shift scale. And so we always use a frequency of a reference peak and the peak of interest. Now, this is a small separation. It's kilohertz at max. And of course, the frequency, the Larmor frequencies we're talking about are megahertz range. And so this is a parts per million. These are parts per million differences. And that's the origin of the chemical shift scale. Wow, having trouble advancing that slide. And so, like I said, it's really, you know, these nuclei, these NMR active nuclei are excellent reporters on the local chemical structure. And here I'm showing the chemical shift ranges for the proton. It has a relatively narrow chemical shift range. It's a first row element, not a lot of electron density around it. But as you can see, we get separation, and we're able to characterize the environments based on the ranges they typically occupy here. Now, carbon 13, the NMR active isotope of carbon, it has a much wider chemical shift range, second row element, more electron density around that, and it's much more sensitive to this. Unfortunately, it's not a very receptive nuclei compared to the proton, so we tend not to measure it directly in most NMR metabolomics. But there are indirect ways to get it at natural abundance, get its information. And so I also want to point out that most metabolomic studies are carried out in aqueous environments in the solution state, that is. And we typically won't see the acidic or alcoholic or amine type protons. So our chemical shift range is a little tighter. And that's because these trade with water, with the solvent. And we also have to actively suppress the signal from water because, as you can imagine, it's quite large. So to illustrate some of the properties, the sensitivities of these protons to their environment, I thought I would make a simple example with some alcohols, the first three alcohols in solution, and discuss some of what we see in an NMR spectrum. And so for the simplest one, methanol, we see a single peak corresponding to these methyl protons. It sits a little past around 3.3 relative to the reference. In moving up to the next higher alcohol, we see a dramatic shift of that methyl peak to a lower chemical shift value. It's more shielded now that it's not, these protons are not attached to a carbon that's directly attached to the electronegative oxygen. We also begin to see some fine structure that I'll talk more about on the next slide. Moving up again, extending that chain, we see another shift to a more shielded position. This one's not as dramatic as the first. And we also see, again, more splitting patterns. This fine structure is very important in NMR. And then finally, moving up to the isomer of propanol, we're back down to two signals, and that's because these methyl protons, they have to, on average, experience the exact same environment. And so they contribute to one signal, which is split, this fine structure. And the methine is up here with the highest D-shield value in the spectrum. And so as you can see now, as we move up to more complicated molecules, the features you have to identify a molecule become more. We go from a single peak to not just two peaks and two chemical shifts, but also these splitting, this fine structure. And as you can see, these isopropanol and propanol, their isomers, they have the exact same match, yet in NMR, their spectra are quite easy to tell apart. So that splitting we saw is called J coupling, and it's basically this proton-proton coupling, or the coupling that we're seeing here between protons is transmitted through bonds. I'll spare the details on the origins of that, but these splittings also report on the number of neighboring protons by this N+1 rule. And the peak separations, this coupling in Hertz, is not magnetic field dependent too. And so as we move to different fields, this will remain a constant. One more thing I just want to point out is these simple patterns get more complicated. If, say, for example, these protons on the CH2, if there's some kind of bond, if there's kind of restriction and bond rotation and things like that, where they don't sense the same environment on average, that'll lead to more complicated splitting patterns. And so once again, when we look at an NMR spectrum, in this example, ethanol, we have two chemical shifts, and then we have these characteristic J coupling values that are going to be the same between them. We also see J coupling to other NMR active nuclei. It's a little harder to spot in a natural abundance spectrum because carbon's only carbon-13 is only 1.1% natural abundant. But if we have a sufficiently intense peak in the spectrum, we can see these. And so I want to show you this example of carbon-13 we call satellites in acetate. So when we have splitting due to another atom, we call them satellites. And as you can see, I've had to clip vertically the main peak for the carbon-12 acetate. But as we go down towards the bottom of the peak, we can see this splitting here, which is a two-bond J coupling between carbon and hydrogen. And it's being split by this neighboring atom having carbon-13. And if these protons are directly attached to a carbon that's carbon-13, they split even more with these characteristic values. And so these are not only diagnostic and indicate the coupling, but we can also integrate these peak areas and they'll report, in this case, natural abundance. And so if you think about carbon-13 isotope tracer studies, these proportions would change if something became labeled. And so we can, in one peak here, in this case, in methanol, not only get information about the directly bound carbon, but also neighboring carbons as well. And so it's site-specific labeling. So talking about integrating peaks, so NMR is inherently quantitative. And what that means is that the integrated areas of these peaks, and I want to stress integrated areas, it's not simply peak heights, it's actually integrating the full area of these multiplets, is directly related to the number of nuclei that contribute to it. And so if we look at ethanol in this example, if I integrate these peak areas here and here, the ratio of those will be 2 to 3. And so this is adding yet another layer of information. So we have chemical shifts, we have J couplings, and we have ratios of protons consistent within a molecule. Now, in a mixture where the concentrations vary, if we have a calibrant that's added at a known concentration, and of course we know the number of protons it has, we can calculate the concentration of any other metabolite we identify in the solution. Now, the spectra that I've shown so far are pretty simple, single molecules in water. In a real spectrum, we can have thousands of peaks from hundreds of compounds. This complicates identification and quantitation, but we do have tools to get around that, and we can disperse these signals from a 1D into multiple dimensions by adding pulses to our sequence and putting incremented delays. We can get more chemical shift disbursement and more information from these. And so they take longer. And so they're not generally measured for every sample in a metabolomics analysis. You might selectively choose some or pool samples. Also, you lose some quantitative accuracy in these 2D experiments. Higher dimension experiments are also possible, but when you get into those, you really have to consider labeling. So I want to show some common 2D experiments that are used quite often in NMR and show you some of the information we can get from it. Because we use these to do structure elucidation, and so of course these are good to identify molecules in complex mixtures, especially if we already know where the chemical shifts are going to be or should be. So the homonuclear case, I'm showing what's called a total correlation spectroscopy spectrum. The diagonal axis, since this is a homonuclear, it's essentially this 1D spectrum that I have projected on the top. And what these off-diagonal peaks report on is, for a particular chemical shift, it's telling you all the neighboring protons in a chain of J-coupled nuclei. And so even though this doesn't J-couple directly to this, it does to this. And so you can go down unbroken chains of J-coupling and map out what we call spin systems. And these are very diagnostic and used to identify unknowns or to confirm ideas of other metabolites in complex mixtures. We can also do heteronuclear two-dimensional experiments. The one I'm showing here is called the HSQC. And what this one does is it correlates a particular proton chemical shift to the chemical shift of the carbon that it's directly bound to. And so now we have the even further chemical shift dispersion because of carbon-13's wider chemical shift range. And so, as you can see, we get a nice spread of signals. Also, capitalizing on those J-couplings, again, we can actually phase peaks in the spectrum to tell us whether the pH correlation has an even number or an odd number of protons attached to it. And so that can be very helpful when you get in areas where there can be some ambiguity and you're trying to make an assignment. And so what about carbons that don't have a proton bound? There are experiments we can do to get at those as well. This one's called the heteronuclear multiple bond correlation experiment. And what it does is it correlates the chemical shift of a proton, not with the directly bound carbon, but with the neighboring carbons 2, 3, and even sometimes 4 bonds away. And as you can see, it will pick up the chemical shifts of quaternary carbons. And then in the schematic, I moved down here just to show that you can continue to go down the chain and you can begin to see how this is a useful way to construct molecules from these characteristic chemical shifts and correlations. So now that we have an idea of what NMR is and some of the things that we can do with it, I'm going to discuss a little bit about the typical workflows. Now, in metabolomics, the most common experiment is the 1D proton experiment, and that's because it's a very sensitive nucleus. It's everywhere. So experiments are fast, and no matter what other analysis you might do, you're going to start here. But regardless, in any study, the sequence of events is much like any other analysis, except for there's differences when it comes to the sample prep as to what's necessary. And then we have to get it on the instrument, acquire the data, get the signal into a form that's easier for us to interpret, and then decide how we're going to go about analyzing it. So on the topic of sample preparation, NMR is quite tolerant of wide-ranging sample conditions, can handle low and high pHs and even high salt situations. A lot of times, sample prep can be as simple as filtering a solution or adding a biocide and a calibrant and a small amount of D2O or deuterated water if we're in aqueous solvents, which you are a lot. I'll say something about that later when I get to the instrument setup. We don't need to do prior separations of aqueous samples. There's no need for chemical derivatization. And if we're working with tissues or extracts, you know, the common techniques like MPLEX, we can take those samples and use those in solution state NMR, and we do that quite often as well. These are pictured here are some sample tubes that we use to measure NMR, and they're typically 3 or 5 millimeters, and volumes are around 150 to 600 microliters. But there are specialty probes and specialty tubes for ultra low volumes and especially salty samples, and so we're not limited to this. Now, I want to mention something about a particular strength in not having to do extensive sample preparation by showing you an example from a study I'm currently involved in where it's an eco-metabolomic study. We're studying methanogenic bacteria in sediments. And so we get these pore waters by taking wet sediment, centrifuging it, and pulling it off the top. We can take that directly, add a calibrant, put it in an NMR tube and measure it. And then if we want to get more identifications and we need to concentrate, because as you can see, these two are on the same scale, same gain. And so this is their real relative intensities. Here I've exploded the scale so we can see some of these metabolites like ethanol and acetate and acetone. So we can screen these samples pretty much as is and get at this information. And if we need to, we can go ahead and concentrate later. And in this particular case, whenever you do stuff that dry down, you're going to alter the concentrations of low molecular weight alcohols or acetate. And if their concentrations aren't altered significantly, they can end up being undetectable, especially for like acetone and ethanol. And so we're able to screen samples and send them through additional processing steps because NMR is non-destructive. And so we can send it down other pipelines or do the screening. Like I said, if you've ever had an MRI, then you were the sample in an NMR experiment. And so the technique itself didn't hurt, but the bill might have. So getting it on the instrument, how do we generate such high magnetic fields? Well, they do that by putting a high current into a superconducting coil, and this coil only superconducts at very cold temperatures. And so this sits in a bath of liquid helium and it's surrounded by a vacuum space. And that vacuum space is surrounded by a bath of liquid nitrogen, which is surrounded by another vacuum space. And so you can see if you've ever walked on the floor of an NMR instrument lab, the reason these instruments are so large is because they're big thermoses. Now the probe that the sample goes in is usually inserted in the bore of the magnet, and it holds the sample at the strongest part of the static field. And it has coils to transmit and receive the radio frequency signals that are generated in the hardware over here. Now, the signals that we're reading back from the sample are extremely weak, and so that they have to be amplified. And you want to do that before thermal noise can accumulate, so you're not amplifying that too. And so the amplifier is usually located closer to the probe. In practice today, we use what are known as cryoprobes. And what this does is it keeps the detection circuitry, the coil, and the preamplifier are housed in here together, and they're kept at cryogenic temperatures. And so doing that, we boost signals noise by 3 to 4 times, and that amounts to a huge improvement in sensitivity, and it cuts down on experiment times as well. So when it comes to setups, I've listed some things that we need to do to get things going. With the modern hardware, modern software today, routines to automate these steps are very, very robust. So a lot of times it's a matter of just entering your metadata, deciding which experiments you want to run, and hitting go. Since I did mention that D2O earlier, that's for the log. Basically, the instrument has a separate channel that monitors the signal of deuterium, which is also NMR active, of course. It does not interfere with our detection on the proton. But what it does is it monitors that because it's sensitive to changes in the magnetic field, or so very small drifts in the static magnetic field. These are in a feedback loop, and so they'll put currents through a field coil to basically keep the magnet locked at field. Because if it drifts, then your lines are going to spread out. And so that's what that's for. Acquisition times, it really depends on your sample. If you have decent concentrations, it can only take a few minutes. But as you saw in that pore water sample that I showed earlier, the one before lyophilization, we might measure one of those for a couple hours. 2D experiments, you can count on them always taking longer than the corresponding 1D. But in decent samples, even a 2D experiment, there's especially homonuclear ones, they can be quite fast. But then again, these can also extend to several hours or the better part of a day. Like I said, it always depends. So once the signal is acquired, we need to do some things to convert it into a form that is more interpretable by us. And so I've listed some steps that we do to do that. I think some of these steps will be highlighted in more detail later today during the data analysis or data processing talk. So when it comes to approaches to NMR data analysis, like many of the other approaches that have been presented, we have untargeted approaches or targeted approaches. And so in an untargeted approach, you would basically divide that spectrum of chemical shifts into various regions and perform statistical analysis to see where significant changes in particular regions are occurring. And that's opposed to targeted analysis, where you'd be identifying individual metabolites and their concentrations, and then performing analysis on that. And so in the global profiling or untargeted approach, there's some additional processing steps that you would need to take, and decisions have to be made about how you're going to align spectra and binning. These are not trivial choices, and there's many approaches to them. And so I just want to point that out. It's not always a simple and straightforward decision process on how you're going to form that binning. These are best suited for high throughput analysis, where you have studies where you have hundreds of samples, lots of data coming through, and critically, where you have good control over sample preparation and measurement conditions, because what you don't want to be picking up are differences in sample prep, but differences in whatever the biology is, whether it's disease versus control or depth in an ecometabolomic study. And so, of course, these we see a lot of use in these in biomarker identification and medical metabolomics. Target analysis, on the other hand, seeks to identify and quantify metabolites from a predetermined list, and these would have existing spectral entries. In the previous talk, you mentioned that this is through a hypothesis-driven approach. In practice, most of the analysis we do on smaller scale studies, where the statistical methods and the binning approaches aren't as robust, maybe because, number one, we don't have the number of samples, or we don't have the control over the sample conditions. We do an untargeted analysis that looks very much like the targeted analysis, except we extend this to trying to identify all the features in the spectrum after exhausting our database search. This is actually the most common approach we use here. Very rarely are we only interested in a select list and not anything else. So when it comes to the databases, this is a weakness of NMR. The databases that we use to identify compounds are much less populated than those for MS, and I mean much less. And so I was trying to get a hard number on the number of unique spectra, and when I say of different metabolites, I mean standards. There's certainly at least 800, could be 2,400. It depends on how they count, and I'm not sure if some are being double counted. There are some open access NMR spectral libraries for metabolomics. There's actually quite a few. Some of the most popular ones are the HMDB or the BMRB, but like I said, there are more. These databases usually have some limited spectral search feature or function. And then others have, like Colmar, they have quite robust and extensive search functions for 1D and 2D data. And I'll show you an example of Colmar search coming up. And so automating steps in metabolite identification, NMR is great for the high throughput analysis data acquisition. When it comes to automating metabolite identification from spectral databases, in addition to the limitations based on them not being as populated, chemical shifts can be sensitive to pH, concentration, and types of cations when it comes to salts and temperature. If you measure things at different fields, you'll see small adjustments, especially where multiplets are concerned, because they'll have slightly different chemical shift positions. Those are predictable, but you still have to predict them when you're approaching the analysis. And of course, signal overlap in 1D spectra can make things complicated. There are still several approaches that seek to automate or make semi-automated a lot of steps in the entire workflow, from study design all the way up to biological interpretation. Of course, the NMR side being focused on the acquisition in processing, especially in the identification. And you can custom tailor if, for example, you're looking at a specific biofluid like urine, where the samples, you have a lot of control over the sample conditions and that sample similarity. You can make robust and automated tools for targeted analysis. The thing is they don't necessarily translate to other types of biofluids or other types of studies. Here at EMSL, we use a program by Genomics. It's a commercial program, and it has a proprietary spectral database of 338 compounds, and these were measured at a variety of fields, as well as a pretty significant range of pH, and that makes metabolite identification a little easier. It uses a visual pattern matching approach to identify metabolites. And when you fit these visual patterns to the peaks, they're also calibrated so that when you get a peak fit, you're also getting a calibrated concentration. You can also import databases like the HMDB and use those chemical shifts for additional metabolite identification, but those are not calibrated if it's outside their database. And so if you want to do quantification, you have to go offline and do some manual integration in those cases. So just to show you an example, a quick little example, in line with the acetylturious cells that were mentioned in the Agile Biofoundry, they asked us if we could measure some NMR on some of their samples. And so we took a look at the intracellular metabolites that were extracted in MPLEX. And since these had been analyzed by GCMS, they were interested to know if we could pick out the cis and trans isomers because the GCMS couldn't discriminate between those. And so we measured the spectra. This is the spectrum here. The signals are pretty low for these compounds. I'll show you. So they're in the database. We have multiple features to make these identifications because these two isomers have very different chemical shifts for both of the protons and proton regions. And so we go in, we match from the database the peak shapes. This one's a little obscured with overlap. And as you can see, the features, the shape here and the J coupling here fits quite nicely. And our residuals, this green line is the residual from the fit, is quite smooth too. And so we have confident identification. We also have quantification. And both of these happen to be in the extract that we studied, less than 10 micromolar, but still able to detect them quite nicely. If we need to confirm, if we need to make confirmations of low level metabolites like this, we always spike in a standard, which I've shown here. And if we have more concentrated metabolites, we can make use of 2D NMR. And I show an example of this on this slide. So I took that same sample, I measured a 2D HSQC spectrum, so I'm getting proton and carbon chemical shifts correlations. Colmar has an online server. You can take your peak list and you can put it through their program. They have a variety of 2D experiments that you can use in their analysis, and their database is actually up to 700 compounds right now. And so when you do this, you get a visual return on the peaks that showed up in your peak list and how many were assigned to a single compound or multiple compounds or to no compounds. And then you also get a chart talking about the uniqueness. So once again, NMR, you have multiple features to identify, with the exception of those that only have one peak, you have multiple features from which to make identifications. So you get uniqueness scores on how many matched. And in this particular sample, we were able to go back into the Genomics program, and in our 1D spectrum, able to quantitate 11 of these 18 that were confirmed. Or 11 of 18 were confirmed in this case. And just with additional untargeted profiling, we're able to come up with 29 more metabolites. And if I spent more time on it, I could probably make many more assignments too. So on my way out, it's actually with the delay, I'm not sure exactly how I'm doing on time, but I'm sure I'm probably almost out of it. So I wanted to just showcase some articles in metabolomics or omics research that have an NMR angle where NMR had a particular advantage to the study. And this first one that I'm showing here, just kind of showing the versatility of NMR. It was real-time in organism NMR metabolomics in a whole organism. So they took C. elegans, these worms, they fed it carbon-13 enriched E. coli. And as you can see in here, they put the live worms in an NMR tube, added capillary with some D2O for that instrument lock, and measured 2D spectra. And from these, they were able to look at the changes in concentrations over time of several metabolites. In their TOC graphic, they just showed glucose, but there were other metabolites as well. And they were able to map out different glucose production pathways under different conditions where they knocked out these catalytic subunits. And so this is a pretty cool application of NMR in metabolomics, I think. Very, not completely unique as well. You'll find other examples of whole organism real-time metabolomics. The next three articles that I wanted to highlight were EMSL user projects. So the outside users submitted proposals to EMSL, and my colleagues in these cases collaborated and produced some really interesting studies. This particular one was looking at microbial interactions in hydraulically fractured shells. And so these microbial communities didn't exist there. They were inadvertently added there by the process of fracking. And so these bacteria under these enormous selective pressure of high salinity, low oxidation, high pressure, seeing how the strategies that they used to survive in this environment made for a really interesting study. And as you can see in this network, certain metabolites were identified. Glycine butane, for example, was not only an osmoprotectant, it was also being used by this Helen bacteria for energy. But they were able to map out all these interactions, and NMR metabolomics played a key role in this. One of the reasons it was so advantageous to use NMR is these hydraulically fractured shells, these produced fluids, are extremely saline, they're hypersaline. And so you can imagine cleanup in these being quite a mess, but we have salt tolerant cold probes, and we were able to measure these metabolites and help map these out. So NMR highly tolerant of quite a wide range of conditions. This other one, another one that my colleagues here at PNL collaborated on with outside users. This one's of a human interest, looking at this autism spectrum disorder, where they implanted microbiota from children who had autism spectrum disorder in mice, and then they saw behaviors that were typical with the disorder in the mice, in the germ-free mice that they had implanted these in. When they looked and did metabolomic study of the colonic fluid and during using both GCMS and the GCMS was using the colon and the serum. They did NMR of the colonic fluids. The thing I want to point out here is 21 identifiable metabolites that were significantly changing. Only one was identified in common between both methods, and it turns out that two that had been cited as deficient in patients with ASD were used as a therapeutic in mice with autism, and they saw a reversal of behaviors. And so one of these was taurine, which was only picked up in the NMR. And so the reason I want to highlight this one is it shows the complementarity of the techniques, and how if you have access to both approaches, why not use it, increase your coverage, and pick up more things, because they both have strengths and weaknesses. And on that, the last one I highlight, and I'll get DOIs and links to all these and put them on the drive in the folder where we can upload those for everyone. But this one had to do with determining the structure of unknowns and combining the power of both mass spectrometry and NMR as a filter. So they would get a retention a retention time, a peak and unknown, generate structures of all isomers, predict tandem MS and NMR spectra, and then look at the experimental spectra to weed out all the false positives on those. And so that's another interesting application that I'd like to point out. And so in conclusion, I just want to reiterate that NMR is a highly versatile tool. In metabolomics research, it's capable of providing expanded and complementary methods for metabolite coverage with other orthogonal techniques. Although it's relatively less sensitive than other tools, it will report on the most abundant metabolites without any instrument method bias and is inherently quantitative. Its ability to tolerate wide-ranging conditions, and sometimes extreme sample conditions, minimize sample handling losses or any biases that can be introduced by sample handling. And sometimes, as in the case of those hydraulically fractured shell produced fluid, sometimes it's the most accessible technique for a given problem. And as we saw again, the fact that it's non-destructive permits multiple analysis on one sample and even in vivo metabolomics. And so it's also fully capable of providing very detailed 3D structural information and can be combined synergistically with mass spec for the difficult task of determining or identifying unknowns in complex mixtures. And so with that, I'll conclude, and I'll be happy to take any questions.
Okay, thanks, Robert. I think we are over time by 2 minutes. So maybe one question, then we break for, so let me see which one. We'll respond most of the questions, but there's new questions coming out on Discord right now. So there is like question from Andrew, like, are there certain compounds that are better suited for NMR versus MS? And he's just pointing that there's a lot of examples of organic acids. I'm
sorry, I was having trouble hearing you, actually. Is that a question that's written in the chat that I could read?
Yeah, it's on the Discord right now.
Okay. Yeah, I'm sorry. Could you repeat the question? I'd have to open Discord.
He was questioning about what type of compounds are better suited for MS or NMR, and he also says there's a lot of example with organic acids. Would the study looking at those type of molecules perhaps be better suited for NMR versus MS, or they're equal?
So NMR is with NMR, what you see is what you get, and there's no like instrument biases when it comes to detecting compounds, and so like we don't have to rely on particular molecule's reactivity to be able to ionize. If it's in there in its solution and if it's enough concentration, sensitivity is really our limit, and so if it's there in sufficient concentration, we can detect it. NMR is great for picking up, like I showed when we looked at those pore waters, because we don't have to do sample analysis. A lot of times we'll get those low molecular weight alcohols or ketones and other molecules that would otherwise volatilize during other sample handling steps.
Okay, cool. So I think we're 5 minutes over time. We're supposed to have a 15 minutes break. So I think we're going to do a 10 minutes break and come back with Bill's presentation. There's more questions for you on Discord, Robert. If you can look at an answer there, that would be great.
Okay, we'll do. Thank you.
All right. Okay, so we'll be back at 11:30, 10 minutes from now.